What's up guys, Dr. H here with Tutorial Zone, and today we're going to go over glycolysis. What is glycolysis? Glycolysis is simply the way we use glucose as energy. So the thing is that when you eat carbohydrates, there's starch and there's all these other forms of sugars involved, but they are broken down by various enzymes like the salivary amylase and whatnot into the end product that we are concerned with today, which is glucose. But before we go directly into glycolysis and discussing what happens in those reactions, we first of all have to understand a few basic things about glucose. So, for glucose, it has three fates. The first fate that we're going to talk about is number one, glycolysis. So, it will be oxidized for energy. And in the process of this oxidation process, we're going to yield a product which is known as pyruvate. Now, what's the difference between pyruvate and glucose is simply the fact that glucose is a 6-carbon molecule, whilst pyruvate is only a 3-carbon molecule, which even makes much more sense for the name of glycolysis, meaning the lysis part just means splitting up. So we're just trying to split up a 6-carbon molecule, into a 3-carbon molecule for energy. So the other fate of glucose that we're going to discuss later on as we go on in this series is that glucose again is oxidized to form what is known as ribose 5-phosphate. Sorry, the board is small, I can't really finish writing it. So when it's oxidized to this ribose 5-phosphate, it's a very important reaction in that, meaning the ribose 5-phosphate, it is the backbone, or rather the starting point of formation of nucleic acid synthesis. Nucleic acids, in layman's terms, you can just mention DNA. Everybody knows what DNA is and how important it is in the human body. The other fate of glucose that we have is that it can be stored. And it is stored in the form of glycogen. So we are now talking about glycolysis. Glycolysis happens in 10 steps, but it has two phases. This first phase we're going to discuss right now, and I know it's probably confusing because you're seeing these cartoons on the board, you don't know what they mean. I'll further on explain as we go on in the lecture what just everything stands for, and I hope it makes sense at the end of the lecture. So the thing is that there's the first phase, because first of all you want to utilize energy, you want to produce energy, and in the process of producing energy you have to invest something towards it. So this is the first phase where we're investing, and it's called the preparatory phase, or this is the investment phase as I like to call it. So in the investment phase, the first step we're going to talk about is how glucose enters the cell. So glucose will enter the cell via what is known as the glut transporter. So these transporters, there are 4 transporters: GLUT1, GLUT2, GLUT3, and GLUT4, which I'll further explain more in another lecture where we're talking about transporters and how they work. So once the glucose is in the cell, the glucose, like we say, this is a 6-carbon molecule. So we have 1, 2, 3, 4, 5, and 6. So once it's inside the cell, we kind of want to keep it inside the cell. And how do we do it? This is where these guys will come in. So these guys are enzymes, and enzymes, they catalyze a reaction. So this is a substrate of this reaction, and we're going to have a product. So these enzymes, their names are glucokinase, and hexokinase. So you're probably wondering why I drew glucokinase in this way and hexokinase in this other way. So this is just a simple way of remembering that glucokinase is predominantly found in the muscles, whilst hexokinase is an enzyme found in these other cells of the body. So what does this enzyme do? So what this enzyme does is that it'll get an ATP molecule. So an easy way to remember is that we're going to have this guy, which is an ATP molecule. ATP meaning adenosine triphosphate. And I'm going to represent this triphosphate because it means three phosphates, just like this. We have one phosphate here, another here, and another phosphate here. So what this guy does is that it will grab onto this phosphate, and once it grabs onto this phosphate, what happens next is that one molecule of this phosphate will leave here and it will go here to this glucose molecule. This is just a structure representation of what glucose looks like. So this is the glucose. So glucose, 6 carbons again, carbon number 1, 2, 3, 4, 5, and 6. So the phosphate molecule will go onto the carbon number 6 here. So when it goes onto this carbon number 6, one thing you have to take note is that ATP is being converted into ADP because it is losing one molecule of phosphate. So it's no longer triphosphate, it's now adenosine diphosphate, di meaning 2. So it will now be with 2 phosphates, right? So the phosphate that leaves, the representation will now be in the structural form now. The phosphate, which will come here, will now be here. So that's the first reaction. And when it comes to the carbon number 6, the name is going to change. It will no longer be glucose only. And because it is attached to this number 6 carbon atom, it will now be known as glucose 6 phosphate. And again, when you're writing down in this format, we're just going to put 1 ATP will be used up from ADP because it lost the phosphate. So this is the second reaction that happens, oh, rather, the first reaction that happens. So again, we're going to put it here. If suppose this is our first carbon atom, so it'll be 1, 2, 3, 4, 5, 6. So this is where we're going to have the attachment of the phosphate. I hope we're clear on this point. So again, the purpose of us converting glucose into glucose 6 phosphate is we don't want it to leave the cell. I hope it's clear up to now. So the next part we're going to go over is the conversion now of glucose 6 phosphate into fructose 6 phosphate. So the enzyme responsible is this guy over here. I drew this guy in this form just to remember the name of the enzyme. So this is a 6-sided. So 1, 2, 3, 4, 5, 6. And what do we call a 6-sided polygon? A hex. So the name of this enzyme is known as hex, meaning 6-sided. Hex, os, denoting the fact that this is a sugar, isomerase. So an isomerase, it is an enzyme that only has the job, okay, even if you just see the way I drew it, it is sort of like doing this thing. So what it is doing, it is that it will pick up this carbon molecule and rearrange it, right? So it will rearrange it in the form of fructose 6 phosphate. So what's the difference between fructose 6 phosphate and glucose 6 phosphate? Is that fructose 6 phosphate is a 5 carbon ring sugar, and glucose 6 phosphate is a 6-sided carbon ring. So when you count even here, 1, 2, 3, 4, 5, 6, 6-sided ring. But for fructose 6 phosphate, and again, our phosphate will still be attached to the number 6 carbon, which is here. So it will retain the name fructose 6 phosphate. Even when you count the size, it's 1, 2, 3, 4, 5. I hope we understood up to this point. So the next reaction that happens is that, and this reaction, one thing you have to take note is that it is a reversible reaction. So meaning that it can go from glucose 6-phosphate back to fructose 6-phosphate and vice versa. So the next reaction we're going to have in this step, in a simplified version, still going to remain the same, but when you look at this form, you'll be able to understand and appreciate what is just happening in this process. So after this process happens, we're going to have another enzyme that will come into play. So this enzyme, again, when you just see the way I've arranged them, these guys with their hands up like this are the kinases. The kinases are grabbing other molecules and stealing a phosphate from those molecules and attaching them somewhere else. So this is what's going to happen again. So again, it'll grab onto an innocent ATP molecule. Like we said, ATP having 3 phosphates. 1, 2, and 3. So after it grabs this, like again we said, 1 phosphate will leave. But where will it attach to now? So when 1 phosphate leaves, it will go and attach itself at carbon number 1. So what does this mean now in the long scheme? So again, we're going to remain now with the ADP, meaning it only has 2 phosphates. So what it means now, in the grand scheme of things, it is now that this fructose will now have 2 phosphates attached to it, at carbon number 1 and carbon number 6. So the naming of this compound is something I really want to go over again, because you can either say fructose 1,6 biphosphate or fructose 1,6 bisphosphate. So the difference is just something minimal. So here's the thing. If you say fructose 1,6 fructose biphosphate, it will simply mean that there's 1 carbon atom here and both phosphates are attached to the carbon atom. That's simple. But when we say bisphosphate, it means a simple way I like to remember this is that when we're putting bisphosphate, the S here, S will denote space. It means there's a space between the carbon atoms. So it means that it is attached at carbon number 1 and at carbon number 6. I hope it's clear up until this point. So this is now how it will look. So after it loses the phosphate here, it goes and attaches here. And again, at carbon number 6, there's another phosphate here. So, like I said, fructose 1,6 bisphosphate. I hope we're clear up until this point. So we're now going to go over the second last step that's involved in the investment phase. So and again, here never forget to put ATP to ADP. Right? Clear. So now this is the favorite cartoon I'll put here. When we are now looking at this cartoon, when you look at this enzyme, you know, he's wearing the head wrap over his head. He's looking like a karate master and he's doing the chop. And it is for this reason why the same way he is looking right now, it's the same action he kind of does in the reaction. So this guy is called aldolase. So what does this karate master do in this reaction? So what he simply does is that when he chops, he will split the molecule, because this is a 6 carbon. So he will split it into 3 carbon and 3 carbon. So split it into 2 halves. So here we have 1 phosphate here and another phosphate over here. So the reason why I even drew them in different colors here is that it's signifying the 2 different 3 carbon molecules are going together after this reaction. So when it comes here, in this simplified version, so it's going to chop up until this point, splitting it into 2 different molecules. What are these molecules? So the first molecule we're going to have is dihydroxyacetone phosphate. And then the other one we are going to have is glycerolaldehyde 3-phosphate. So when we're looking at it in this manner, this guy will split it just up until this point, so down right here, into these two molecules, the glycerolaldehyde 3-phosphate and the dihydroxyacetone. So the difference between these two molecules is that the glycerolaldehyde 3-phosphate has an aldehyde group attached to it, and the dihydroxyacetone has a ketone group attached to it. That's just a simple difference. But in this case, the one that we're going to want in this particular reaction is the glycerolaldehyde 3-phosphate. It's the one meant for a reaction. And how are we going to get it again? So again, we're going to have another guy that's going to come in. I drew this guy because he has three sides, 1, 2, 3. And I don't know if you remember from the last time, when you see them like this, means it's an isomerase. So what it's trying to do in this reaction is rearrange it into the form that we want, which is the glycerolaldehyde 3-phosphate. So the name of this guy again, if you can just predict from the other one, we said the hex ose isomerase. So this one, because it has three sides, I'm going to say tri-ose isomerase. And what is this guy going to do? This guy is just simply going to convert the dihydroxyacetone, which is this one here. Sorry, phosphate here. Not phosphate here. So it's going to convert dihydroxyacetone into glycerolaldehyde 3-phosphate. And this process is reversible. So the point when we have glycerolaldehyde 3-phosphate is the end of the preparatory phase or the investment phase. It is called the investment phase because we're investing 2 ATP molecules. At the first reaction here, catalyzed by this guy or this guy, glucokinase or hexokinase to form glucose 6-phosphate. And the other one is at this point, we're going to invest another ATP, and this guy is going to be used up by phosphofructokinase, this guy here. So this marks the end of the investment stroke preparatory phase. We're now going to go into the payoff phase.